Hello, everyone. My name is Yannick Dalpuy. I'm the Scientific Affairs Manager for Bionano, and I'll be your host today. And I would like to welcome you to this exciting webinar featuring Laila El-Khattabi from the Cochin Hospital in Paris and Alexander Ocean from the Radboud Medical Center in Nijmegen, the Netherlands. Dr. El-Khattabi and Ocean will present us with their new results from their comparative studies between classical cytogenetics and Bionano for the identification of structural variants in constitutional or cancer sample. I would like to remind everyone that this event is interactive. Please ask questions during the presentation, and to do so, simply type them into the Ask a Question box on the side of the screen. You can also share on social media by clicking the tab on the left side of the screen. Before getting started, I would just like to take a few minutes to present you with the Bionano technology. NGS still struggles to resolve complex variants or variants buried in repetitive sequences. To answer that, Bionano is based on sequencing, but just like you would do with a microscope by looking at metaphase spreads, Bionano brings you the true structure of a genome. We do this by visualizing ultra-high molecular weight DNA molecules using a scanner we developed, the Sapphire, similar to what you would do with karyotyping. Let me show you how it works. The Sapphire workflow uses the chips to linearize DNA. We have kits for DNA extraction and labeling, and our own free analysis tool. Let's break down the workflow together. Our technology relies on the direct imaging of ultra-long DNA molecules. That's why we developed a fast protocol where we lyse the cells in solution. Then we use a nanobind disk that will tightly bind the DNA molecules and protect them from any mechanical stress. And now you simply wash it, elute it, and it is that simple. The protocol is fast and perfectly automatable. Note that FFP samples are just not good enough to be used with Bionano, as the fixative will slightly degrade the DNA molecules. We can do this with fresh or frozen material, blood, cultured cells, fresh or frozen tissue, or tumor biopsies for as low as 10 milligrams. One main difference with the long-read sequencing is that our molecules have an average length of about 400 kb and go all the way up to megabases. Once we have the megabase-sized DNA, the next step is to label it. Here, an enzyme will recognize a 6-base pair motif and attach a fluorophore directly to the DNA. The DNA will also be counterstained with an intercalating dye. There's no nicking and no probe involved here. It's a single enzymatic reaction, labeling a 6-base pair sequence that appears every 6 kb on average, and the pattern is unique throughout the genome. So at any time, we know where the DNA molecule belongs in the genome. Then we linearize the DNA. We use electrophoresis to pull the DNA into nanochannels. The array looks like this, what you have on the top, hundreds of thousands of channels in parallel. The stretched DNA is the bottom channel. So the blue signal is the counterstained backbone, and the green you see are the labels we attach to the DNA all throughout the genome. The principle is similar to karyotyping, but at a much greater resolution. And once again, the pattern is unique throughout the genome, so we know where the molecule belongs in the genome. Now the instrument will cycle the molecules into the channels and image them over and over again. Cycle them until you get the massive yields you need to analyze complex human genomes. You can go anywhere between 100x to 1600x coverage for exactly the same price, the only variable being the time you run the instrument. Once we collect these images, the software will perform de novo assembly of the molecule, meaning we align every molecule we imaged just based on the label pattern. The de novo aspect is important, as when you work from a highly rearranged material, you do not want to force the assembly to a reference that would not make sense here. And then we compare to the reference. In this case, the reference is in green. We will align our maps de novo assembled to the reference and look for differences. In this case, you see a deletion because the space between our labels decreases. And of course, all the knowledge from the reference still comes along, meaning that we know where the genes are because the pattern is unique throughout the genome. So that way we can detect each major type of structural variation automatically, genome-wide, starting at 500 base pair. We can detect insertion, deletion, repeat expansion. We detect tandem duplication inversion automatically at 30 kb and above. Translocation and inversion are really hard to detect with sequencing, and we are very good at it. We also developed a specific pipeline for complex heterogeneous genomes that leverages the tremendous coverage and looks at SVs at the single molecule level. And this is crucial when you have a clonal population like in leukemia. This tool is complementary to the de novo assembly pipeline I just presented and allows you to detect all structure variants 5 kb and above automatically, genome-wide, down to 1% allele fraction. And that's a game changer, and no one else can do this. You can visualize our genome in our browser called the Bionano Access, and you can see a circus plot like this, where each of the chromosomes are shown in concentric circle. Different types of SVs are present here as dots. The purple line in the middle are translocation between each of the chromosomes. If you just click on any of these events, you can zoom on the map that just popped up on your screen, where you can see the de novo map aligning to each chromosome. Here it's a translocation. We can even show the molecules that we image with our system. But on top of that, all the SV call will be filtered to a control database. So you can just filter only somatic events, rare events that are not present in the control population. And of course, the annotated SV call can be exploited as a standard VCF file. Optical mapping has raised a lot of interest in the human genetic fields, whether it is to modernize old cytogenetic methods, such as karyotyping, to quickly identify actionable SVs in cancers, such as NTRK fusion partners. But it also led to numerous discoveries of fusions never described before. I invite you to visit our website or to download the vignettes you will have on the side of your screen. But I cannot wish of better results to illustrate what Bionano can bring you than those the story of our two speakers are about to present to you. So without further ado, is it my pleasure to welcome Dr. Allele El-Khattabi from the Cochin Hospital in Paris and Dr. Alexander Oyshen from the Radboud Medical Center in the Netherlands. Dr. El-Khattabi is an associate professor in cytogenetics at the Descartes University and Cochin Hospital in Paris. She's in charge of the laboratory of non-invasive prenatal screening and fetal aneuploidies for Parisian public hospitals. She's also involved in the diagnostics of chromosomal abnormalities related to developmental and reproductive disorders. In addition, she leads research at Cochin Institute on gene transcription and how this function is impacted in case of chromosomal abnormalities. Dr. Oyshen is an associate professor and group leader at Radboud Medical Center. His research group is called Immunogenomics, with a 50% affiliation each at the Department of Internal Medicine and Human Genetics. In the last years, he has shown that application of novel and disruptive technology will allow new scientific insight and rapid translational into clinical and diagnostic practice. For now, please, Dr. El-Khattabi, the floor is yours.
Thank you, Yannick. Hi, everyone. Thank you for being here, and thank you for presenting me, for giving me this opportunity to present our work. So, for today, I'm going to present the results of our collaborative study between my lab in Paris, my French collaborators in Lyon and Clermont-Ferrand, and the Radboud Genetics Center in Nijmegen with Alexander Oyshen. So let me just give you, please. First of all, just to precise that part of the Radboud reagent cost of this study was covered by Bionano Genomics. So let me just set a few things first. As you know, structural variants account for a large part of variation in the human genome, and this leads to phenotype variability, but sometimes it leads also to diseases, and especially developmental and reproductive disorders. So in the past years, we've all known the expansion of clinical genomics thanks to the development of new technologies, among which sequencing, high-throughput sequencing, and this led to new discoveries and an increased diagnosis rate for these diseases. But the detection of structural variation is still challenging, and that's why karyotype and chromosomal microarray analysis are still used, at least for reproductive disorders for the first one, and developmental and malformative disorders for the second one. But as all of us know, karyotype has a very low resolution, estimated at 5 to 10 megabases, and microarray do not detect balanced rearrangement nor provides information on the location of an aberration in the genome or its orientation. So we thought to test this new technology because we thought it may improve the diagnosis of structural variation, in particular balanced ones for our patients. So we joined our forces with Nijmegen, and we started the study on known chromosomal aberrations. So this was led on patients who were referred to us for reproductive disorders or intellectual deficiency, developmental disorders, or multiple congenital anomalies, for which the current standard of care first-line test is karyotype or DNA microarray. So whenever these techniques detected a large SV or CNV, and we had leftover material for a patient, then we just included that patient and to realize the genome mapping using Bionano. So here is a description of our samples. 85 samples were included. As you can see on the left side of the screen, the indications were quite diverse, corresponding to what we have in a clinical lab. And on the right side of the screen, you can see the different cytogenetic investigations that were carried. So in about 1/3 of cases, we had karyotype, another third CMA analysis, and the last third both of these. In total, we had 114 aberrations. 50% of them were balanced and the other 50 unbalanced. And the distribution of the kind of aberrations was very diverse again here. As you can see, we had even isochromosomes, ring chromosomes, complex aberrations, but most of them were deletions and duplications, as well as translocations as expected. Here are the first overview of the results. So this is the average number of calls that we got. In the dark blue shows the total raw number of SVs. The marine blue in the middle shows the SVs after filtering, in particular with the population frequency, and the light blue bar shows the numbers after overlapping with genes. So sorry because it looks like small, but in total we had just a handful number of SVs after filtering, 70 in total, which were distributed between different kind of aberrations. So just to set the principal results first, we got 100% concordance except for aberrations within unmappable regions of the human genomes, of course, centromeres, as well as P-arm of acrocentric chromosomes. And here are 3 examples of those, where you can see in the red font the breakpoints that couldn't be located. So now I'm going to show you a few examples so that you can see how it looks like. The first one was a karyotype from a man showing a Klinefelter syndrome. And as you can see, it was a 47XXY karyotype. So genome imaging showed the aberration using the CNV call system. So here is the circus plot. In red, the region corresponding to sex chromosomes, and here is the zoom in of that region. So you can see from the CNV trap, which is shown here by the blue plot, that you have coverage on chromosome X that is twofold higher than the coverage on chromosome X and as high as the coverage on autosomal chromosomes next to it, which indicates that it's XXY karyotype. The second example is an example of a 4 megabase duplication that was detected by CMA here. And here is the result from Bionano, and you can see shown in red gain duplication on chromosome 19. And when you zoom in, here again on the CNV call, you have the green plot that shows that there is a gain in this region, and this is shown as well by the SV call. Okay, so same here, and this is a smaller CNV, 500 kb on chromosome 8, and the CMA result here from SNP array shows the deletion, and here again with Bionano result, you can see by the red arrow a loss or deletion on chromosome 8, and again a zoom in, and the blue plot, which corresponds to the coverage, shows that there was a loss at this region. So here is the representation by structural variation, and you can see in the lower side of the image, the green map corresponds to the reference, the blue one corresponds to the sample, and the labeling shows that there is a loss at this region. Here again, a translocation, but I'm gonna skip the translocation maybe since we have a lot of examples of translocations and we might be short in time today, but I'm open to questions to discuss about it later on. So let's go to another example that shows an inversion on chromosome 3. Here is the karyotype of this woman. On the right side, you can see a zoom in of chromosome 3. The right one shows a paracentric inversion on the Q arm of chromosome 3. Here is a simple circus plot view again from Bionano that shows that there is an event on chromosome 3, and when you zoom in, you can see your molecules. On the patient map track, you have 2 molecules, well, at least 2 maps that show there is an inversion between these 2 regions from chromosome 3. Below of it, you have this detailed, and this is a 23 megabase inversion. This example was from a little girl who showed growth retardation and developmental delay, for which a karyotype showed a marker. And Bionano can show you with the circus plot here. So this is the circus plot only for chromosome X. And you can see there is an event between the centromere and the Q region of chromosome X. And if you zoom in into the genome map of this patient, you can see here again that there is a little map that shows a junction between the left side of the chromosome and the right side of the chromosome, and this corresponds to the marker that is a ring chromosome. So now I'm going to take a little time to show you in detail some other examples that represent for me additional potential input of this technology to diagnosis in genetic diseases. So the first one is an example where this technology can allow for genotype-phenotype correlation. So let's see, this one is a little boy who was presenting intellectual deficiency and other phenotypes. And the karyotype showed apparently balanced translocation between chromosome 9 and 17. So the image represents a result from Bionano, and it shows on the upper line the chromosome 9, on the lower one the chromosome 17. These are the references, and between them a little blue box that corresponds to the patient map. So let's zoom in. It will make it easy. So again, the blue track corresponds to the patient track, the green ones to the reference. The upper green is chromosome 17 here. The lower one is chromosome 9. And you can see that the blue line maps both to chromosome 9 in the bottom and chromosome 17 on the upper side. And we also indicate the genes that are involved in this region from chromosome 17. And you can see that the gene cancel1 would be disrupted. And the haploinsufficiency of this gene is already known to cause Coulon-de Vries syndrome, which leads to intellectual deficiency and facial dysmorphism, which was consistent with the patient phenotype. Another interest of using this technology is to allow for the detection of cryptic balanced aberrations. So here is an example of a circus plot from Bionano again that shows a translocation between chromosome 7 and chromosome 10. Here is again the zoom in, same than previous one. The blue line corresponds to the patient map, the green one to the reference. The upper green is reference chromosome 7, the lower one is chromosome 10. And here again, we can see the translocation between the Q arm of chromosome 10 and the P arm of chromosome 7. Here is the corresponding karyotype, that just to show you that this is very hard to see as a translocation. So on the right side, you have the zoom in of the karyotype, chromosome between the chromosome 7 and chromosome 10, and the red arrows indicate the derivative chromosomes, which are very hard to see for the translocation. Again, Bionano can also provide information on the orientation of the duplicated segments, and here is an example of this. This is a result from CMA that shows the duplication on the P arm of chromosome 8. Here is the result from fish analysis. So I've circled the both chromosome 8. The white circle indicates the normal chromosome, and the red circle indicates the rearranged chromosome. And you can see that the green and red label, which correspond to two back probes, are face-to-face, if I can say, in the red circle chromosome, which is indicative of inverted duplication. So when you look at this case using genome mapping, genome imaging with Bionano, you can easily see both events. So on the CNV call track, you can see terminal deletion, which I didn't talk about, and duplication. And this duplication, you can deduct from the SV call track that it is inverted. And interestingly, although breakpoints of the duplication were located within segmental duplications, this event was detected. We can also reveal and unravel complexity behind apparent simplicity. What I meant by this is such cases with here apparently simple, although three-way translocation between three chromosomes, chromosome 3, chromosome 13, and chromosome 5, as shown on the karyotype below. But when you have the results from genome imaging with Bionano, you can see from the circus plot between chromosome 3 on the right and chromosome 5 on the left that there are much more events than the ones that were expected. And here is a zoom in on chromosome 3 that shows a lot of events, inversions, deletions, that were indicative of chromosynthesis. And I will end my examples with this one. So this is a case of a translocation between chromosome X and chromosome 1. The breakpoint was, we were not able to map this breakpoint even using NGS. And genome mapping with Bionano helped us for this case, since here again, you can see the middle track, the blue line from the patient that maps both to chromosome 1 and chromosome X. And this disrupts a gene called NLG-NA4, for which haploinsufficiency is responsible of ID-ASD, and our patient had moderate intellectual deficiency. So this allowed also a diagnosis for this case. So to conclude, our study revealed a high success rate using this technology with 100% concordance with chromosomal microarray analysis, 100% concordance with karyotype when breakpoints lie within mappable regions of the human genome. And this very large molecule analysis allows to pick up small and large SVs, and even in cases where breakpoints lie within segmental duplications. So we expect significant benefits to diagnosis. First, genotype-phenotype correlations for a lot of cases with balanced rearrangement, or as well as unbalanced. New discoveries in unsolved cases. The workflow is quite simple with this unique technique that could replace 3 of the techniques for now. The easy technical and analytical process is very appreciated, with no bioinformatics analysis, no overwhelming calls after filtering, as we saw, and this is in adequation with the routine analysis process. The other advantage of not having genome sequence is that we will have less incidental findings, which is important in case of investigations for reproductive disorders, for example. And to me, at least, although Robertsonian translocations and centromeric breakpoints are not detectable for now, we expect a high balance of benefit to risk, which would favor the adoption of this technology to our current investigation tools portfolio. We are even, but maybe we will discuss about these for people who are interested in this technology. We think that few cell counting could be just a complementary test to add to it. So please follow us. The publication is to come by 2 postdocs, Monterey Tuomo and Cornelia Nivelin, who have been working hard on this project. And please just allow me to thank all our collaborators at Radboud University Medical Center, as well as my French collaborators, in particular Caroline Schut-Bollard in Lyon, Céline Richard in Clermont-Ferrand, and my colleagues at Cochin Hospital, Jean-Michel Dupont and Aziza Lobar, as well as all other clinicians and genetic laboratory that were involved in these studies. Agence de la Biomédecine for funding part of these experiments, and the Bionano team because they helped us a lot with the analysis. Thank you.
All right, I had to unmute myself. So thank you very much, Leila. I think we agreed that I directly follow up upon that. My name is Alex Huyschen, and as Yannick kindly introduced, I work in Nijmegen, the Netherlands. So I'll directly follow up upon Leila's really nice talk, and then we decided to have the Q&A right after my presentation. So I'll just try to show you a little bit of the follow-up work we've done next to the constitutional study that Leila just presented, and that will be on some novel discoveries of structure variants in rare disease cases, and that is in contrast to the cases that Leila presented where we knew that the aberrations were present. However, we refined several of those as Leila has just shown. And in addition, I will also show you how we can use Bionano for comprehensive somatic SV detection, in particular in leukemia. And with that, just again to make sure, Bionano supported us with some reagents throughout this study. My first slide of the presentation is if I just have to explain how Bionano works to cytogeneticists, they immediately get thrilled. While some people that mainly worked on sequencing may still have some issues in understanding all the benefits and where we are with Bionano at the moment. So the easiest example I can usually give is it actually is very similar to karyotyping, and I think that's why I termed it some time back next generation cytogenetics. So it still is looking at large chunks of chromosomes. So just imagine you would be a cytogeneticist. This would be chromosome 1 that you would look at. Imagine you could just zoom in to each and every cytoband and just have a massive zoom in for that. And many bands, up to 1,000 bands per chromosomal bands. You wouldn't be used to that, but that is in fact what Bionano can now offer. And as such, it still maintains all the other benefits that cytogenetics can give you, but it just is cytogenetics on steroids with something like 500,000 bands compared to 500 bands that you usually would get. So that basically would be cytogenetics with a 10,000-fold improved sensitivity while still maintaining the benefits. It still is a genome-wide analysis. It still gives you the positional information, and in principle, it also allows you to look at single molecules. And we'll come to some of these advantages in the next slide. So this is just an overview slide of what we've done. Also, we are still quite new to the field of optical mapping by using Bionano. So we basically have the instrument in running for a little bit more than a year. But due to the COVID-19 crisis, obviously, we haven't been as productive in the last 3 months as we planned. But this is basically what I can show you today is the output of the first year of use Bionano in-house. So as Leila has nicely shown, we roughly added half of those samples that Leila presented today. Since then, we've done a few more samples for constitutional cytogenetic aberrations. So those by and large are samples that had a known aberration by karyotyping fish or microarray. And we just asked the simple question, can we find those true positive events back? And as Leila has shown, it seems to be relatively easy. We directly hit 100% concordance. In addition, still talking about constitutional, so germline aberrations, we couldn't resist to also try this novel technology on some of our unsolved rare disease cases, and I'll show some of these examples. So we totally run 50 or so of those samples belonging to 20 projects, and so far I would consider 5 of those 20 solved, or we at least have very interesting leads, and I'll present 2 of those today. And ultimately, when we saw this technology, we thought this may be really good for leukemia because their cytogenetics, or actually a mix of all different cytogenetic tools, is still the gold standard. So we also compared 50 leukemia samples to the routine workup that they usually get. And I'll show you both of these examples today. Just one high-level summary, what we usually do. So for the constitutional samples, we usually find with just generating 80 to 100x genome coverage data. That usually is used to build a de novo assembly of the genome, and then we have a combined SV and CNV calling from that de novo assembly by comparing it to the reference genome. That is all the work that Leila just very nicely presented. The samples we have used in the first year is a wide mix of sample inputs. Just as a reminder, we can't use readily isolated DNA, but we use this very special protocol that Bionano has optimized. And we use various blood types, bone marrow aspirates, but also various cell lines so far, and that worked very nicely. Different from the constitutional analysis, the leukemia data that I'll show you later, obviously there we need higher genome coverage to gain even more sensitivity because the main target there would be to look for somatic, hence acquired aberrations. So we usually go for a 300x genome coverage there. And then instead of running a de novo assembly, we use the latest tools released by Bionano, and that is a so-called rare variant pipeline that is specialized for somatic events. But again here, similar to what you do for constitutional aberrations, it makes use of two different algorithms that allows an SV and a CNV calling. And I'll give you examples a little later. But for now, let me start to show you some of our research cases. So when having the instrument, we directly thought, okay, this may be nice to tackle some of the unsolved rare disease cases that we had collected over the years. Some people may anticipate that this may unravel some structural variants that remained refractory to NGS detection. Or some other people even refer to those events that those may lay in the camouflaged parts of the human genome. If I just give you the three main categories of rare disease patients or initial approaches to that, those usually were families or cases that already had a known linkage region in the genome. Equally, we studied some cases with a classical recessive disease where only one pathogenic mutation was discovered, and we tried to use Bionano to find the second missing allele. And then we had some very severe sporadic cases where we speculate that the disease-causing mutation may just be a hidden de novo structural variant that we couldn't detect otherwise. All those samples were, the vast majority is obviously collected in-house in Nijmegen, but we're also involved in two large-scale studies, the SolveRD and the Cross-Omics Initiative, and obviously we're open to other collaborative studies, and I'll show you some of that as well now on the next slide. Because our first example is a collaboration with my former colleague, Roland Kuiper, who now works at the Princess Maxima Center in Utrecht, also in the Netherlands. Roland and his team and collaborators are interested in a rare but very severe condition called atypical teratoid rhabdoid tumors. These are very rare but aggressive tumors that are usually diagnosed in childhood. Those usually present as severe brain tumors, mainly affecting the cerebellum. And there is already a known cause, and those are mutations in SMRT-B1. And it is known that 1/3 of cases present with a germline mutation in SMRT-B1, while 2/3 of sporadic cases present with a double hit somatically. In the case I present today, there was a suspicion for a genetic predisposition, so a germline mutation. So Roland and his clinical colleagues were already studying this family for 2 years or more, because in this family, unfortunately, a very young girl already died of her brain tumor and the ATRT condition. However, the parents still had another wish for another child, so they were curious to understand whether there would be a recurrence risk due to a germline mutation or not. In the tumor of this girl, there was evidence that SMRT-B1 was affected based on immunochemistry. However, it was still unknown whether it was a sporadic somatic event or really a germline event. So for that purpose, they used germline DNA and offered diagnostic assays by Sanger sequencing, MLPA, and even exome sequencing, but no germline mutation was found. Then Roland's team also did genome sequencing by standard short-read Illumina sequencing and still was unable to find, oh, sorry, I was distracted by the chat, but we're still unable to find a mutation in this gene. So then they sent germline material and EBV cell line to us in Nijmegen. We performed Bionano-optical mapping, generated 300x genome coverage, and did very simple standard filtering. And you see all those filter settings on the right-hand side. What it basically comes down to, if you just do very simple filtering and only focus on the structural variants that have never been seen in any control, we end up with 35 rare structural variants in this case. And then obviously, we zoom into the most important locus for this disease. One of those 35 rare events hit the SMRT-B1 gene, as you can see here. The upper bar shows the gene tracks. The green bar, again, similar to the previous presentation, is the reference genome with the suspected label sites for the Bionano labels. and the blue bar represents the de novo assembly. And there you can see that a small portion is inserted. If I zoom in a little bit further, then you can directly see the individual molecules that support this event. So there's sufficient molecules really showing reliably that this must be an insertion. And if you look at the size, what you would expect based on the wild-type marker distance, you would expect that these two markers have a 5 kb distance, but in the patient, heterozygously, we saw an insertion of 2.8 kb, and we even saw an additional label. Other than that, obviously, we don't have any sequence context for this aberration. So in our view, this very likely is the germline mutation that was looked for so much time by so many different technologies. It is most likely inserted between introns 1 and 3 of the SMRT1 gene. However, that's probably the only downside. So far, we don't have or had at that stage the exact inserted material. But then initially we thought, oh, but it should be easy. We just do a long-range PCR over that locus. But for still unknown or not fully understood reasons, none of the PCR approaches actually worked. So we speculated that maybe repetitive DNA that may be entered here. And actually, we just finished those validations. And what we just did, because none of the targeted approaches worked, we simply did PacBio sequencing. So we used our latest PacBio Sequel 2 and produced a HiFi genome, so very high quality CCS coverage genome, 8-fold coverage with a median read length of 20 kb. And this supported an insertion exactly where we expected it, and in this case, an intron 1 of SMRT1. This is shown by the purple bar, and that's 2 of those insertions. I'll just show you the next field here. If you click on the inserted sequence, we may get a feeling why it was difficult to validate it. There is a very repeat-rich region on the left boundary and the right boundary of this inserted material. And if I tell you now that this sequence, especially at the boundaries, but the entire sequence, really mimicked a retrotransposon, in particular an SVA retrotransposon that is inserted. So by having now base pair evidence, we're really sure now that this is the germline aberration that we were hunting for. What remains to be seen, or what is still an open question, I should say, is how can an intronic insertion lead to a loss of function of the allele? I mean, we have evidence on the protein level of the tumor that that is really the case. However, how this may disrupt splicing is still currently looked for in Roland Teiper's research lab. But obviously, intriguingly, it is now to speculate how many more of those jumping retrotransposons do we have in the human genome, and how many other rare disease cases may this explain. That is very intriguing, obviously. Okay, fast moving to the second case I wanted to show you today. We obviously study a lot of sporadic cases with severe intellectual disability. That's what we also did here. So this is a sporadic case born to healthy parents. And in this particular case, we've done everything we had so far in our genomics portfolio. Classical follow-up for such a case is CNV-microarray exome sequencing, then usually followed on a research site by genome sequencing, and in this particular case, even by PacBio long-read sequencing. Most of that is published data for this particular case, and the long-read data are just submitted. And unfortunately, none of these previous technologies found anything disease-causing in this case. So we speculate that maybe this one case may have a hidden structure variant that escaped the detection by all the other methods. So what we again did is we just did optical mapping for this case, did a de novo assembly. If you look at all the structure variants, that's close to 6,000 or 7,000 or so. If you then again just do very simple filtering by filtering for events that are rare, so never seen in any control genomes, in this case 200, then we end up with 30 to 50 or so structure variants genome-wide. But in this particular case, because we suspected a de novo event, we also included the parents and just clicked one extra filter saying, show me only the variants that are not present in mom and dad. And that's what you then see. Here is one predicted de novo deletion. So then just click on this one event. We end up in a very complex region of the human genome, which is indicated by the orange bars at the bottom. This is one of the most complicated segmental-duplication regions on 17q21. So what we saw in this patient is actually that there was a paternally inherited duplication overlapping with the NSF pseudogene number 1, But in addition, just partially overlapping with this duplicated sequence, predicted de novo deletion, actually a quite large one, 121 kb. So you could directly wonder, why wasn't that picked up by any of the previous sequencing-based assays? And the answer may lay in the complexity of this region. It really fully overlaps with a highly complex segmental duplication. On the next slide, we see our validation experiments that we just recently did. So designing primers on the mutational allele only amplifies the mutated allele in the patient, but we don't see any hints in the father or the mother. This amplicon was then sequenced again by PacBio sequencing, but in this case, not genome sequencing, but rather amplicon-based sequencing that spanned this region, obviously. And on the next slide, sorry, I took that slide out. This amplicon sequencing did two things. It first of all validated the events, but also gave us exact breakpoints based per resolution. And it showed that, in fact, this came down to quite exactly 121 kb de novo deletion, identified by Bionano and now validated by PacBio amplicon sequencing. The gene that is disrupted in this region is obviously very interesting, what that could be. Obviously, this de novo event happened in one of the most complex regions. This is this terminal segmental duplication of the 17q2131 microdeletion syndrome, also known as colon de Vries, as shown briefly by Leila for a different purpose. But within this de novo deletion, the gene that may be disrupted or is likely disrupted by this deletion is a gene called NSF, which is interesting is that this gene has an important brain function and is mainly expressed in brain. However, as already shown in the previous plots, this gene has a pseudogene NSF-P1. And actually both genes together are very copy number variable. Anything between 2 and 8 copies, to my understanding, have been reported in the general population. But to my understanding, there was never a case described with just one NSF copy, and that could be what we are facing here in our patient. So it is far from any evidence, but it is very tempting to speculate that potentially haploinsufficiency of this locus may cause disease. And what actually fostered this further is that there's a very recent report from wonderful research performed in Japan, and this group identified de novo point mutations in NSF1 in patients with specific forms of epilepsy. However, they speculate in their paper that most likely their effect is a dominant negative effect. Nevertheless, this is the first evidence that NSF is a disease gene, so we're now trying to gain extra evidence in our patients to see whether the only de novo event that was now found finally by Bionano may be even involved in disease. Right. Now I'd like to switch gear for the last few minutes and just give you some highlights of our leukemia study. For those that are interested in details, because due to the sake of time, I can't give you all the details, please visit our preprint that we put on bioRxiv. To just keep it very short, the beauty of that study is we used 48 leukemia samples to study all their previously known somatic and acquired events that have been identified previously by karyotyping FISH and or CNV-microarrays. And also, similar to the study Leila presented, we had 100% concordance for all the structural variants that were at least present in 10% or more variant allele fraction. This is just the overall average QC for all the 48 samples. The only two aspects that are important here is that we generated 280x coverage of all those 48 genomes on average, and the average molecule size is 262 kilobases, but individual molecules are much, much longer. This is the overall averages of structure variants that we find per case, just to give you a flavor. These are the results of the so-called rare variant tool, or rare variant pipeline, so that is different from the pipeline Leila used in the study. And that on average shows us close to 1,500 structure variants per sample. However, of those, only 44 on average are rare events. In addition, we can benefit from a potential overlap with genes, and then we're only facing on average 25 or 26 structure variants per case. In addition, we can use the CNV tool that you basically get on top for free, and that shows us on average 9 gains and 50 losses. However, these numbers may be inflated by some of the highly complex cases that I'll show you in a second. Just to give you a flavor of what we usually see in these leukemia samples. So here we had a case with a known balanced translocation. On the left-hand side, you see the genome-wide circus plot. You've now seen plenty of those. And on the right-hand side, you see a zoom in to chromosomes 11 and 14. That was the known balanced translocation in this case. If I just click on the purple line on the right, that directly guides us again to the map. Again, the sample map of this leukemia sample is in the middle in blue. And it maps to chromosome 11 reference and the chromosome 14 reference on the bottom. And the predicted breakpoint indicated by the purple bits is directly showing us that this is a balanced event fusing most likely the IGH and the CCND1 locus as just circled here. I should say that usually the breakpoint resolution comes down to just a very few kb for most of the cases. Because those are somatic events, we can actually just click on the sample map, and we actually then directly see how many molecules support this event, and we directly have a flavor for how high the fraction of affected alleles is. This is just a summary of 3 additional translocations that we found. So there were 3 CML cases involved in this proof of concept study. They all carried the hallmark aberration of CML, namely a Philadelphia chromosome between chromosome 9 and 22, which obviously leads to the BCR-ABL fusion. On the left-hand side, you see the ABL breakpoints in all the 3 samples. So you see 2 different breakpoints in ABL1. And on the right-hand side, you see the BCR breakpoint. Again, 2 different breakpoints are predicted based on the Bionano results. In summary, I can say that usually the breakpoint resolution we get from Bionano is much better than what we would get from traditional FISH. Ultimately, we can also start seeing very complex cases in much greater detail than ever before. In these cases, we had 1 sample with chromotripsis of chromosome 8, also shown in the zoom in on the right-hand side, and a monosomy 7. For the sake of complexity, let's just focus on chromosome 8 for now. On the next slide, we see the actual map of this chromotriptic chromosome 8. And what you just have to imagine here, each and every blue fragment is a map of fused pieces of chromosome 8. So in principle, we would now for the first time be able to completely rebuild the structure of this chromotripsis just based on the Bionano optical maps. Final bit for visualization, here you see 1 sample for which we had CNV-microarray results with a trisomy 12 and a smaller deletion of chromosome 13. The upper plot shows our standard asymmetric cytoscan HD array, and the lower bar is the new genome-wide-CNV visualization that is very much array-like that Bionano now presents. And then again, on the right-hand side, you see the zoom in, and actually it's almost indistinguishable from the array results that we're so much used to. Ultimately, sorry for a little mess up on the slide that you now see. That was not intended. Just in addition, because in principle, we only looked at variation that we knew previously, just for fun, we filtered all the rare structure variants identified in all 48 samples. Of all of those, 95 presented with rare interchromosomal translocations. 23 of those were unique calls that led to potential gene fusions. And if I just show you one of those examples, there were several known ones in there, obviously, like BCR-ABL that I've shown you before. But in this one case, there was a fusion that, at least to my knowledge, was never described before, for sure not in this case, but also not in other leukemia samples. And that is a likely fusion between the gene AGBL4 and RUNX1. On the next slide, again, that's a zoom in to the map of the fusion. Again, this is really most likely breaking these 2 genes or fusing them together. Obviously, to people familiar to the leukemia field, RUNX1 is a very known, well-known player. But at least to me, AGBL4 was not ever described in leukemia. But it is interesting that AGBL4 fusions have been described in other cancers, at least for lung adenocarcinomas. That was a known fusion partner for other oncogenes. So this is just to highlight one of these examples. But actually, all the data is online. So if you want to play with our data, all of that is on a preprint, as I've shown you before. Just to wrap this part up, I've shown you briefly some highlights of our study of 48 leukemia samples that have been previously tested by karyotyping, FISH, and CNV-microarrays. They consisted of 37 simple cases and 11 complex cases. And in summary, we find 100% concordance for all simple cases, and we also have full concordance for 10 out of 11 complex cases. In one complex case, we missed 3 aberrations that likely had a variant allele fraction that was just below 10%. Getting that accurately counted is not even so trivial. So as such, we also have additional evidence now that for some of our very complex cases, the events that we anticipated were even more complex. And I dare now to say that genome imaging, so Bionano, is usually much closer to the true underlying aberration nature of the event. Just as a final highlight, one more slide of something very new. We just played with some of the data. As some of you may know, in leukemia, regions of homozygosity are very interesting, or in particular, losses of heterozygosity that happen somatically. In this example, I show you just a plot of all the SV codes along chromosome 1. And you see that we see different codes, some homozygous, some heterozygous codes, if I just go to all the events in this case, including the germline aberrations and the somatic events. But if you go to chromosome 7, this plot really looks different. And the dotted line in the middle just gives you an averaging window. If you look at chromosome 7, then we see a total loss of heterozygosity for chromosome 7q. And that was a known event in this case. This was a large region of loss of heterozygosity. And although it's not supported yet by Bionano as an automatic calling, it now seems to be relatively easy to spot that in the data. And we're quite excited about this extra feature now. So again, look at the data or play with the data if you wish. A lot of the data is given in the supplements for download and tables. And as such, that brings me to my summary slide and a little bit of an outlook. I hope I've shown you, actually, the first part Leila has shown you, we can get full concordance for all the aberrations in the constitutional aberration fields, except for those breaking in the centromeres, obviously. For leukemia, we also generate full concordance 100% once aberrations are in an allele fraction higher than 10%. And for that reason, I mean, I'm just a researcher, but I'm carefully optimistic that Bionano may be the first technology that could completely replace cytogenetics, and as such, that will be karyotyping arrays and FISH. As a researcher, I'm probably even more thrilled about the discoveries we've recently made. So the structure variants that were undetectable by other means are now popping up by the Bionano analysis, and that obviously gives us very interesting leads for our own research. I honestly believe that optical mapping may allow a true revolution of cytogenetics, and that's why I also like to call this next generation cytogenetics, and this is just starting. And then as a final teaser, I think there's some really cool new innovations coming, additional colors, additional labels. Yuval Edelstein's lab is working on really fancy methylation next to structure variants by Bionano. And what was mentioned briefly by Yannick is that we can generate for the same price much higher coverage now, which even allows you to find somatic mutations that are in 1% variant allele fraction or lower. With that, I'd like to thank all my collaborators. Obviously, a massive thank you again to Leila for the wonderful collaboration with her and all her collaborators, particularly for the work shown in my slides today. I'd like to thank Tuomo and Cornelia for their wonderful work over the last year, and many other colleagues from Nijmegen, the entire team from Roland Kuiper in Utrecht, and our funding sources and Bionano for their support. I'd like to finish by telling you that the currently shown products are research use only, and then I'll leave you with citing the genius Sidney Brenner at the end that tells us how important novel technologies are. Thank you very much for your attention.
Thank you very much, Dr. Oysen. We would like to take some time for a few Q&A questions. So Yeah, thank you again for your time and for your presentation. We will ask as many questions as we have time for. We will probably not have the time to tackle every questions. We will get back to you via email after this conference for those that we cannot answer right away. Some interesting questions are regarding the filtering steps and the amount of SVs that are detected in this technology. Can you give us a bit more insight on the filtering step that you used, Dr. Al-Khattabi and Ocean, either for the constitutional side and more somatic detection, and what are the tools that you can use to further filter the SVs that you identify? If you want to start with Dr. Al-Khattabi.
Yeah. Okay. Yeah. Thank you. So yeah, I saw there was a question about this, and my slide on this theme was not really easy to look at. Actually, I've shown a histogram with the means, averages of a number of calls, and someone asked how many you pick up at the beginning, and it's a few thousands. Like the mean is around 5,000 to 6,000 events that are detected. But once you filter out with the population frequency and you pick up only rare events, then this drops harshly down to a mean of 70 events. So we've decided to start with the settings that are recommended by you guys, Bionano actually, and which are based on few confidence scores on the results, etc. And actually, we were really happy with the results, so we stick to it. And as I mentioned to someone from the attendees who asked me, we had to just play a little bit with few filters for 2 cases only in our study, but actually the recommended ones were quite good.
And for Dr. Ocean?
Yep. So I mean, Leila nicely answered it for the constitutional changes. I think there we use very simple filters, obviously, just for rarity of the events, very similar to what people are used to from the microarray field. And we did the very same also for our research constitutional samples, as I've briefly shown you. So usually we end up by just using trivial filters pre-selected in Bionano, and we just have 30 to 50 variants per genome that are extremely rare or unique. So that already is an absolute manageable number. For some particular cases, we can also do a dual analysis, so doing either 2 affected per family and do an overlap, or 1 healthy, one affected per family, and subtract the ones that are presently healthy. And what we do more and more is obviously running trios, so unaffected parents to extract the de novo events. I think we all know that de novo germline structure variants are extremely rare. It is still anticipated, even based on genome data, that there's less than 1 per genome. But if you find one, that may be a more immediate hint towards a novel disease locus. So I think that is a very interesting approach, and it seems that Bionano doesn't make too many false positives or create a lot of noise. So the filtering for de novo seems to work nice and straightforward.
With the ability to also import bed files to also focus the analysis, so you would keep the whole genome information, but also mimic let's say like a fish panel to highlight regions in the genome that can be of further interest for your study. Correct?
Yeah, I can answer that. Yeah, absolutely. So I think Leila showed briefly that how many of your 50 rare events overlap then with genes. That's genome-wide is only just roughly half or 60%. But if you're interested only in the 100 genes that are expressed at a certain moment in a certain region of the body, then you can add that gene list and just do an overlap with that. So in silico filtering for events that overlap with genes is very simple. You just need a bat file.
Yeah.
Great, thank you. A question for Dr. Al-Khattabi. We can get from the comparative study that you just presented that not detecting Robertsonian translocation is an impediment, but still you state that the benefit-risk balance is in favor of adopting the genome imaging into your genetic portfolio. Can you develop on the percentage of patients presenting such disorder translocation in your cohort, and how you intend to deal with such translocation?
Yeah, sure. So for at least for reproductive disorders, Robertsonian translocations are kind of up to half of cases can have Robertsonian translocations, but this is in extreme cases of recurrent miscarriage, etc. The other part of reproductive disorders, male infertility, female infertility, the part of Robertsonian translocations is much lower, but still there are cases. So definitely you would not rely only on genome imaging for those patients, but actually it's so easy, and you can pick up new events. Probably that's our next step in such patients, but it's really interesting to move forward with the technology, but still maintain, at least in our case, we thought about it, and we were thinking of maintaining a minimal count of a few cells in addition to genome imaging, which is quite easy to manage and imagine in a genetic diagnosis lab, of course.
Yeah. As a very informative complementary technique to the
exactly. So I would say genome imaging as a first line test, and the complementary would come from karyotype. Well, not a full karyotype, but at least a cell count. Yeah.
To relieve the burden on the karyotyping.
Yeah. Count few cells. Yeah.
A question for Dr. Ocean. So the conclusion of both constitutional and cancer studies presented 100% concordance with the standard of care on the retrospective cohort, and besides, you identified numerous novel fusions and striking leads in unsolved cases. What will be the next step for this comparative study in your institute?
Yeah, so obviously I'm working fully at the research side, but now we're discussing quite actively with my colleagues at diagnostics what the next step is. And in our view, this is a more prospective study where we would run the standard of care workflow in parallel to Bionano and then compare the outcome of that. So while we still then generate our standard results, I think a relatively modest series of 50 to 100 samples run in parallel would already tell us whether this is ready for more prime time diagnostics. So I think that is something we will probably do now relatively quickly. Yeah, so prospective after this study.
So think that the results on the unanswered cases, the added value on the diagnostic side for the complement of information on classical cytogenetics. We obviously implementing a new technology such as Bionano in routine is a long multi-step process. And can you both of you share your view on what would be a realistically way to implement this technology in your institute? And first, as I understand from Dr. Tabi, rather in the complement of the classical cytogenetics, and can you also discuss the integration with NGS that is involving the same ecosystem and that brings a different kind of information? I don't know if you want to start, Dr. Ocean.
Yes, very happy to do so. So I think many genetic labs around Europe or across the world are used to lab-developed tests. So I think for that purpose, it may be happening that we start using this as we did the first generation microarrays relatively soon, even in a clinical or preclinical setting. So I think we wouldn't necessarily need to wait for an FDA-approved test for the time being, but we could just use it, especially if we convince ourselves as specialists on these diseases that it works and that it is better than the standard of care. And so far, we've shown that it is at least as good, and we certainly see some advantages. So I think that may be straightforward, but maybe that is my research mind that sees things here relatively straightforward. And maybe Leila from a real diagnostic lab has another answer to that. Just to very briefly touch on your question of the combination with NGS, I truly see an opportunity that we can generate almost perfect genomes by combining exactly what you said, Bionano optical maps with NGS, either short or long read data. So I think for the first time, we may get close to an almost perfect genome analysis if you combine these two assays.
Yeah, so there are two main situations, as I mentioned, reproductive disorders and developmental disorders, and the decision process for each is different, right? So for now, at least in Europe, but I think it's overall the same everywhere in the world, reproductive disorders we still rely on karyotype. For developmental diseases, we do have PMA as first line, NGS as second line or first line, depending on every country. We do know the limits of karyotype for the first diseases and the NGS and CMA for the second. So absolutely, I share with Alex the statement that for developmental disorders, it would nicely complement NGS. And we would just skip CMA analysis, definitely, I think, with having Bionano and NGS. For reproductive disorders, the question is as we said, the added value and the newly diagnosed cases, because we know that karyotype is very low, has very low resolution. So, but still, with the limit for centromeric breakpoints, I would say that, again, genome imaging first and complemented by mini karyotype or cell count or whatever you want to call it, but definitely genome imaging has a place to take in both diseases.
Thank you very much. We do have some interesting questions about the COVID-19, and is it possible that people are affected by COVID-19 who may have chromosomal aberration? And this is an interesting question because we actually do have some studies running in China, in Hanover, in Germany, specifically looking at the incidence of the presence of structure variants that may or may not predispose patient to the COVID-19. So we had some information released on our website, and feel free to get back to us to get more information about this. We would be happy to put you in touch with the people leading these studies. I'm just scrolling through this question over there. I'm sorry, there's just so many questions pouring in that it's difficult to scroll through them. Maybe a question for Dr. Ocean. What is the anticipated rates of test failure with leukemia sample, and especially one with bone marrow, from bone marrow?
Yeah.
Sorry.
Yeah. So just the blunt and honest answer is, I think in the first month, we tried some bone marrow samples that were not recommended. We used much lower input than Bionano recommended us to use, and those samples can fail, absolutely. Since then, we're a bit more strict to just follow exactly the QNC, so the quality checks that Bionano gives. And since then, we have almost no failures, I dare to say. So if you use 1.5 million cells, if you use the DNA stabilizer in your sample directly, and that is either bone marrow aspirates or heparinized or EDTA blood, then I think the failure rate is extremely low at the moment. Obviously, that may be different if your sample is shipped around the entire globe. So if the cells are not intact and your molecules are not intact, then you won't manage. So I think as for some of the sequencing assays, here it's even more important to learn the lessons that, basically garbage in would be garbage out. But if you have high quality cells, then I think the chance of successful experiments is very high by now.
Very good. We are way past time for this session. We do have some question about pricing. How much does it cost to run a sample on optical mapping? So the best person to answer would be, of course, to get in touch with our regional business manager that can provide you with all the different information about the three different ways to get Bionano, either through the service, by sending us directly the sample, through a reagent rental, or through buying directly the system. But the pricing starts at $450 per sample. But just get back in touch with us, and we will be happy to put you in touch with your local regional business manager. Thank you again, Dr. Al-Khattabi and Ocean, for your research, for your presentation. And before we go, I would like to thank the audience for joining us today and for all the interesting questions. We didn't have time to tackle all the questions. They really poured in at the last minute, but we will address them, and we will provide you with answers in the following days. Thank you again to both our speakers. This webcast can be viewed on demand after this session, and Labrousse will alert you via email when it's available for replay. And until next time, thank you again, and goodbye.